Hi guys, many of you struggled with the Cherry Coke Lab, both the essay and the picture, so this tutorial is intended to try and help you do a better job if you need to redo it, or try and help you get started if you haven't even begun yet. What I've got here is a Venn diagram. I'm sure you've seen these before. This is where differences go on the outside parts of these circles, and things that are the same go in the overlapping part of the circles. Let's start with what the two different things have in common. They're both distillations, right? They're both using the technique called fractional distillation. And fractional distillation is a separation technique for a mixture of liquids. So both cherry Coke and crude oil are mixtures of liquids. Cherry Coke is less complex than crude oil. Crude oil has hundreds of different compounds in it, all of which are hydrocarbons. So crude oil is a mixture of hydrocarbons. And cherry Coke is a mixture of basically water, sugar, flavoring, and CO2. So the fractional distillation process is a little bit different in the two setups, right? But they both are trying to do the same thing. They're trying to separate the liquids at their boiling points. So separating using boiling point, B point. There we go. And that's pretty much it for the things that they have in common. Some of the structures are similar, and we'll talk about that when we do the picture. But for your essay, you want to start by talking about what they have in common. Both of the setups that you looked at, the one for the Cherry Coke Lab and the one for the crude oil, are designed to fractionally distill a mixture of liquids using boiling point. That's your starting point. That's like your opening sentence. Then you want to say, and they have many similarities in their structures. For example, they both have to heat the substance. So they both have some structure that is intended for getting the liquids to their boiling points, right? Because until you actually heat it up, nothing's going to boil, right? One of the differences between the Cherry Coke setup and the crude oil setup is that the Cherry Coke starts distilling, it's in the liquid stage. We have it in a test tube, and it's all liquid, except for the carbon dioxide, which is a gas to start with. And we heat it up, and we turn it to a gas, and it travels up to the top of our test tube, goes through that little glass bend, and while in the glass bend, it turns back to a liquid. And that's how we're distilling it. Each liquid is going to boil at a different temperature. We've got a thermometer there to tell us what temperature it's boiling at. And so we know when we're getting a particular liquid to boil, and we know when a new liquid is boiling because we get a new plateau. We get a change in the boiling temperature. That's how the Cherry Coke works. By comparison, the crude oil, all the stuff enters the big giant fractionating tower as a gas already. So the furnace, which is sort of to the left of your big fractionating tower, that furnace heats everything up to a gas, and then the gas is released into the tower, and all those gases are floating around in there. And the ones that cool back to a liquid sooner are the ones that fall down in the lower trays. So one of the big differences between the Cherry Coke and the crude oil is that in the Cherry Coke, we're going from liquid to gas, back to liquid. But with the crude oil, we're going from gas back to liquid in our tower. That's one of the big differences. All right, let's look at the different pieces of equipment that are used in these two setups. Okay, on the left we have our Cherry Coke setup, right? So we have here a test tube, and that's where the distillation is going to happen. That's where we're containing our mixture, and our mixture is being heated by our alcohol burner down here. And we have a container to the right which collects the liquid as it boils. And we also have a tube that delivers the boiled liquid. And up here, you see this glass bend right here? That glass bend has a very important function, which we'll talk about in a second. Over on the right, we have, similar to our test tube, right? You've got the fractionating tower, or distilling column, as it's named here. See, right there, distilling column or fractionating tower. And we have a structure that is inside that's hard to see that's called a bubble cap. So they made a little picture over here to show you what a bubble cap is. You might have a different picture that has the bubble caps looking something like this, which is also kind of what they look like from the side view. And we also have these little trays, well, not so little, right? Each one of these gray things is a tray that's holding a liquid. And sort of the orange stuff is the liquids sitting on top of the trays, and the yellow stuff are the gases floating up out of those trays. So then you have, dun, 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 in blue, on the right-hand side, your delivery tubes, right here. And of course, your furnace, which is off camera, right? We don't have it, it's not here somewhere, but you can consider this your furnace. All right, so what structures have similar functions, right? Well, on the left-hand side, your test tube is the same as your fractionating column or your distilling column. That's the location where the mixture is being held and where it's changing state. Your furnace and your alcohol burner obviously have the same function. They're there to heat up the substances. And this tube that's circled in blue on the left has the same function that all of these tubes over here have, which is to deliver the newly created liquids to storage containers. And the green circled item here, where this small test tube is in a water bath, that has the same function as the trays do. That's where these liquids are sort of being stored and then siphoned off. And then probably the most interesting piece of equipment that's the same in both is the glass bend and the bubble caps, which are both there for the same reason. That glass bend is where your liquid turns back to a gas in the Cherry Coke lab. Why does it do that there? Well, glass is cold, and we're getting further away from the heat source. So even though we've turned to a gas, our liquid, when it hits that cold glass, turns back into, our gas turns back into a liquid. That tube is actually called, that glass bend is called a condenser. It's there to force your liquids to condense back into liquids. Guess what? That's what your bubble cap is for as well. Those bubble caps allow anything that's still a gas to escape. The bubble cap is not on there tight. That you can see that in this picture, that a gas can come up and get under and escape, right? But a liquid, if you are at the right temperature, when you hit this cold surface, you turn back to a liquid and drip back down into the tray underneath. So it's serving the same function that the glass bend is. It's a condenser. And that's probably the most important similar structure in the whole thing, because without that, the system breaks down. We need something that forces our substances to change state at the right time so we can collect them. So that's probably the most important one. So you can see that I have 4 different colors, and the 4 different colors represent the 4 different structures that you need to cross-label. But you also need to just label, period. Before you start cross-labeling on your picture, make sure you just label what things are, right? So this is, for example, I'm not going to do all of them, but this right here is your test tube. Very simple, right? And on the other side, this one's already labeled in this picture, is the fractionating column. So you have to have the names of the two things, and then you can do like I did. Instead of drawing a whole bunch of arrows going back and forth, you can just sort of use a color coding system, and you can make little dots in each color, and next to the dots, you can write the function that the two things you circled in that color have in common. That would be what I would do. I would recommend that. So that in blue, for example, I have circled these structures that heat the system. So they're the heat source. And you could do that for each of the things, and all you have to do then is circle the structures circle the name of the structure, or circle the structure itself, or highlight it in a color, and then put this little key up here so I can see what the 4 structures are that you're comparing to each other. Hopefully this has helped you be able to do a better job of structuring your essay and writing your paragraph and doing your drawing. You can use any 2 pictures you want to do your cross-labeling. You can even draw one if you don't like any of the pictures you see. There is also 10 points that you get for turning in your lab sheets. Your lab sheets have questions in them that you had to answer. I'm not grading the data table, but I am grading everything else. So the first page and the next 2 pages, you have to get those questions right to get full credit, and it's 10 points. Okay guys, I'll see you in class.